I was asked today to speak by Stu and Debs, really regarding our work on next-gen sequencing, which we've been doing now theoretically for 6 years. First 2 years we were using the good old GS Roche Junior. I don't know if anybody's used one of those, but they were an absolute pain to work with, for very little sequencing data at the end of it. But since then, we've been using the MySeq. We've used the same library preparation system for both systems. Again, it's not off the shelf like a lot of the kits are nowadays. It's a bespoke assay. So first and foremost, I'll talk you through the assay. I'll talk you through our panel design and how we designed it and what we sequence, because we sequence every case that comes into the department of a particular suspected diagnosis. I'll tell you about our bioinformatics pipeline. And again, this is an in-house system that's been developed by Jan Taylor in our department. I'll tell you about how our panel performs and how we supplement it, because as we heard yesterday from Christine Teedy, that these amplicon-based systems don't work for everything, and so we supplement it with extra testing. I'll show you some of the results and show you how our results bear up with everybody else's. I'll also show you how the panel performs. Again, as I was asked by Stu to talk about how we report things, how we annotate things, I'll also talk about that. I'll also talk, if there's time, about future directions of the department, how we are going to do sequencing in the future, our aims for B lymphocytic, chronic lymphocytic leukemia, marginal lymphoma, and then a larger panel that's probably a year away, but again, some of the advantages of a larger hybrid capture type panel that we're planning. So we use a Fluidon access array, and I don't know if anybody has seen these, but they are a microfluidic system on the right-hand side, if that works. Oh, yeah. Also, on the left-hand side, we load our samples and our reagents. On the right-hand side is our assays or multiplex PCR primers. These two wells here we have, but it's almost like mineral oil, and it's the mineral oil which uses to open and close small microfluidic valves. And basically, the reagents and the samples from this side are pushed across this array or this IFC, and our assays or our multiplex primers are pushed along this side. And so what we have at the end of all of this, with pressure used to actually push samples and reagents across the array, are 2,500 individual PCR reaction vessels. And so our multiplex primers, we multiplex up to 10 primers per well from this side, and our samples, 48 samples. So we're running on a weekly basis 48 samples every single week, week in, week out. The workflow is really simple, really hands-off. Again, the amount of hands-on time from library preparation to actually setting up the sequencing is about 2.5 hours. Again, there's no robotics involved. We use multi-channel pipettes to load our arrays. Here. We load it onto a controller unit, and this controller unit is the system that basically under pressure pushes our samples to their appropriate positions. That takes about, well, it says 60 minutes there. The arrays have changed slightly over the last 6 months. That's actually gone up to 1.5 hours. But again, it's completely hands-off. Once you put the array in the system, it's a walk-away system. It then goes on to not a conventional PCR machine, but this in the middle here is a PCR machine. Again, 2 hours on a PCR machine, and you're amplifying up your multiplex primers, maximally 10 sets of primers in any one ampule. We then transfer it to a post-PCR lab, where we put it onto a very similar or almost identical controller unit, and we harvest it. Basically, our PCR products are pushed in the opposite direction. So for every well where your original DNA sample started, you now have, well, in our case, for the Myeloid panel, we have 361 separate amplicons present in that well. We then perform library cleanup, so we clean it with conventional ampule beads. We then tag the libraries with individual barcodes, clean those up, we amplify those up to attach the barcodes, clean the samples up, and then we add to a MySeq. 24-hour on a MySeq, so actually hands-on time is 2.5 hours. Actual time from preparing, it's a normal working day. You can fit it in a 7-hour working day, and you sequence overnight. We run this system every single week. We run it on a Wednesday usually, and we report on a Monday. So the turnaround time for our 48 samples is usually 7 days. We resequence probably about 20% of our samples. Rather than doing Sanger validation, we actually find it's cheaper and easier to resequence through the same system. So we resequence about 10 to 15 samples a week. And again, I'll explain what that sample set are. So primer design is based on overlapping amplicons. And again, although this is BRCA1, this could be any particular long exon. This could actually be ASXL1, which is a single long exon containing 23 amplicons in our system. We have overlapping primers, so every single base on our amplicon system probably is covered by at least 2 amplicons. So every single base. So if you have a poorly performing amplicon, it's possibly not essential because it will be covered by a different amplicon. Yeah. These should be kept separate. In the multiplex system, obviously you've got 46 different wells, so for something like this, and certainly for something like ASXL1, where you've got 23 amplicons, they can be easily separated over your 48 multiplexers. This is to cut down on primer crosstalk. So if you've got a primer and another primer, say 250 base pairs away, they're going to talk to one another and produce a non-specific product. So it's important that you separate your primers in regards to where they are on a length of DNA. So our panel looks like many other panels. It contains 26 myeloid genes and 2 T-cell genes. It contains all the usual candidates: DNA methylation, chromatin modification. Obviously for MDS, the spliceosome, 4 spliceosome genes are all there. Transcription factors and T53 is omnipresent in all panels. Signal transduction, cohesion complexes, and sort of other modes as well. So it looks like everybody else's panel. You'll probably notice on there that there's a definite glaring omission. CEBP alpha is not on there. As was mentioned yesterday, CEBP alpha works very poorly on any amplicon-based system and probably needs hybrid capture to actually work successfully. So what do we sequence? As I said before, we sequence all comers that come into the lab because we treat this as part of the diagnostic process. Again, all our samples are screened with a fairly rudimentary, at least initially, immunophenotyping assay or a tube and a conventional morphology. Any case with a diagnosis of suspected AML, MDS, CMML, or overlap syndrome goes down the sequencing route. For the first 3 years, we also sequenced cases with uni or multilineage dysplasia presenting as a cytopenia. Again, we've got lots and lots of data on these patients, but we probably currently stopped doing those because of the issues of chip, and that actually finding individual isolated rearrangements probably can't really help you in the diagnosis or confirm a diagnosis of a primary myeloid disorder. We're getting increasing numbers of referred cases which just come through the post asking for myeloid high-throughput sequencing. Probably about 20% of all the cases we actually do currently of this form. So it's patients from all over the country asking for our myeloid panel. So how does it all stack up? So you can do the maths. We analyse 48 samples on a 300 cycle version 2 MySeq or bog standard sequencing kit. We have 28 genes, as I said before, 26 myeloid, 2 T-cell specific ROA and STAT3. That's 361 amplicons in total. If you do the maths, that's 48 patients times 360 amplicons, 17,000 unique amplicons. One normal MySeq run generates about 20 million reads. So if you divide one by the other, we're getting an average read per amplicon per individual patient of about 1,000. So perfectly suitable for what we want to do, and we're happy with that depth of sequencing. So once we sequence, we do our own bioinformatics pipeline. We have onboard quality control on the MySeq, and that's your usual clusters passing filter, your Q30 scores. Again, probably usually around between 95 and 97% of our sequencing reads pass that particular QC filter and then go on down our bioinformatics pipeline. We align with BWA MEM Again, we variant call with VARSCAN2. We do some local realignment with ABRA, and I'll explain a little bit about that later on. And then we push our VCF files and we annotate them in Haemato-Batch. This is an automatic process. Haemato-Batch then generates a series of annotation files, and these are probably more like you're used to seeing with a protein location, a cDNA location, the transcript used, etc. So more like you can actually put in an eventual final report. So a lot easier to understand than a VCF file, which is a base pair change at a particular position on a chromosome. We have an automatic reporting process, and I'll explain that in the next few slides. Again, raw data, FASTQ files, BAM files are all stored offline. They're stored in a facility in Harrogate, an NHS facility, on this Isolon server. Again, all our samples are reported by our in-house database, and this has now got 8,000 samples on there, something like 16,000 individual variants. Again, this, in combination with our HILIS LIMS system, generates a patient-specific report, which is attached directly to the HILIS report. As I said before, high throughput, we can do 1 or 2 runs, or we could even do 3 runs a week on a single MySeq. So again, you've got the capability of doing 150 samples per week. So if I just home in on that small the bioinformatics workflow there, we have 2 filtering steps. We have a technical filtering step which occurs on the VCF files. Again, we filter out any reads with any variant with a read below 100 read depth and a VAF below 5%. Again, we retain all our hotspot regions, again, even if they're below that depth of read. We have a series of known exclusions. So these are recurrent variants that you see multiple times in a sequencing run. So you might see that particular variant occurring 20 out of your 48 times. And again, it's a TAC error usually. So we know what these are, and we find these very, very frequently. So they're technical filters. Again, they occur in the VCF file. I mentioned before, we do local realignment on the VCF files because certain abnormalities, in particular deletions, and there's a really common ASXL1 deletion, 23 base pair one. The BWA alignment program won't pick that up, but it will see something very similar. It will see something very consistent, and it sees 3 separate variants. And if you see those variants, or if the software sees those variants, it knows to realign that particular rearrangement to see if you can find that. And it always does find that. So it actually appears as a single mismatch or a missense mutation, and then a small insertion, but it's always the same. So ABRA is used to do local realignments for these types of abnormalities. Again, we have on our Haemato-Batch annotated files, we do an annotation filter. So we're filtering out at this stage synonymous variants, intronic and splice variants, known polymorphisms, i.e. this will go down through a dbSNP database. And we also do our QC filtering here. So Picard metrics, we've talked about the QC filtering. Actually on the MySeq, these are internal metrics, and Picard will pick up every base. It will look at every single base, the number of bases passing 100 reads, so that's one metric. The number of absolute variants is another metric. The actual DNA concentration is another metric. So all of these are actually metrics which we use, and if it fails 2 out of the 8 metrics, then that is a failed sample. Failures, we probably see a failure one sample every couple of runs. So it's quite rare, and usually you can pick up what the problem is. It's usually poor quality DNA. So we aim to put 50 nanograms of DNA into each sample. If we fall below 10 nanograms, we know those samples are going to be poor, and they're going to throw up lots of variants. So how does the assay perform? These are our 28 different amplicons. You can see they're quite varied. We have amplicons such as MIPL here, where we have one single amplicon that's going to cover your hotspot regions, S505, W515. We have a single NPM1 amplicon, picks up the 3' end of exon 15, exon 12, sorry. AXXL1, 23 amplicons, as we said. It's a single exon, so these are amplicons spread across a single exon. big players are over here. TET2. TET2 probably has 63 amplicons. Again, we have flaky genes. STAG2 is a bit flaky, but in general, all of these, even though we have well-performing amplicons and poor-performing amplicons, all of them, on average, will amplify above 100 reads. Again, SRSF2, you can see here, we're looking at a single hotspot, but you can see we've got multiple amplicons at base also. Because it doesn't perform very well, we've thrown 4 or 5 different amplicons on it in a view to try and get the best coverage we can. So how does our panel perform in regards to everybody else's? If you look at the 3 subgroups we tend to concentrate on, AML, MDS and overlap syndromes, you can see that TET2 is omnipresent. The mutations are present in all 3 subgroups. FLT3 is AML specific, both the ITD and the TKD. We find things that are more specific for MDS, SF3B1, not surprisingly, particularly in cases with ring sideroblasts. SRSF2 is also common in all 3 subtypes, as are the rest of the spliceosome genes, and again, they're a very important prognostic group to actually identify. Again, down here you'll see that we don't look for FLT3 ITDs by next-gen sequencing for all the reasons that were mentioned yesterday. Large ITDs are very, very difficult to actually align, even with pull-down strategies. So we use fragment analysis to actually identify those as an extra technique. So supplementing our panel, as I've mentioned before, CEBP alpha, very important prognostic gene, is not covered particularly well by any amplicon-based system. So we use a Sanger-based system. Again, we know that's not ideal. It works in our hands, it works every time. It's a 4 amplicon-based system to cover the single exon CEBP alpha gene. And we also use conventional fragment analysis to look for FLT3 ITD. So again, those 2 assays I run alongside our high throughput sequencing assay. So the data, so what do we show? Well, we've been sequencing on this system with this particular platform since 2014. As we said on the original title, we've sequenced 8,000 unique samples since that time. Between 2014 and October last year, we actually clinically reported 2,500 samples. We found nearly 6,000 reportable variants in these 2,500 samples. And again, our panel and pipeline was UCAS accredited in March 2017. Since October last year, we've been using a new pipeline, a V3 pipeline, which again is the pipeline I've shown you, which uses Hallam batch and our own reporting software. And again, in this time period, we've clinically reported 720 samples. We found nearly 2,000 variants. 940 of them are unique. As I said before, we resequence samples which fall outside our reporting threshold, so between 5% and 20% VAF, or between 100 and 500 reads, we'll resequence those patients. So what types of abnormality, and we're talking about reporting now and annotating, what types of abnormality do we actually report? Well, from those samples from October to present, the 720 reported cases, the nearly 2,000 variants we reported, 1,300 of those actually were annotated with a cosmic ID. So these were our hotspot region mutations, our known classical mutations. A further nearly 500 mutations were likely pathogenic, and by those they were a frame shift or a nonsense mutation resulting in a truncation. So we were fairly confident that they were likely to be pathogenic. And then the contentious area, which I will go on to talk about, are variants of unknown significance. We are currently reporting these, and in that time period we've reported 188 of these. Again, I'll explain in a couple of slides' time why that's actually quite important. We think that's probably a step we're going to go back from. We're probably going to stop reporting those. Again, these are almost exclusively missense mutations. we put a threshold for those, reporting threshold of they have to be present at a VAF above 10%. So this homes in, this gives you an example of our annotation using Hallam batch. So this particular case, we've got 150 entries, VCF entries from our, our alignment. We put that through Hallam batch, resulting in the same 115 entries, but in a more user-friendly base. We filter with an in-house Python script, so basically all this is done automatically. As I said before, we retain hotspot regions, so we've got 160 hotspot regions, so if they're there, irregardless of the depth of sequencing, we will pull those out, we'll keep those. And then we have our 42 recurrent exclusions. These are the things we were talking about that occur very frequently in your sequencing. So they are basically PCR artifacts usually. We also remove at this stage synonymous variants, intronic splice variants, and known polymorphisms derived from SNP databases. And we access numerous SNP databases. That 115 has been now whittled down by our filtering processes to two variants, a DNMT3A, I think it is, and an IDH2 variant. So that then goes through our reporting pipeline, so those variants, along with a unique HMDS number, which is what we use to tie it all up, then goes onto our in-house reporting database, so our two variants there, loaded into our in-house built database. This is connected to a web-based reporting application, and this was all designed by Paul Glover in our department. For each run, you can see this is a particular run. This is this year's run, that's run 22. That will generate a whole series of tick boxes. So these are individual samples. You can go tick on an individual sample, and that will generate this reporting format. Again, on that format, you can open that format up. We go to the, you can open that format up and look at individual variants. Obviously, they've got cosmic IDs and they don't have a DB SNP identification. We'd be quite happy reporting those. On here, we've actually got a CSF3R variant, which is at 50%, been reported in DB SNP, hasn't been filtered out. Again, we filtered that out manually. So for that particular patient, we've actually got 4 variants. We've chosen to report 3 of them. That report then is generated and goes directly to HILIS, our reporting system. That matches up patient demographics information along with the patient ID number, and again generates a report. On the second page of that report, you've got your evidence, i.e. how many times it's been found, whether it's been found somatically, etc. So, back to the contentious issue of variants of unknown significance. And what Dave said in his talk yesterday is, if you put anything on a report, it is likely to be acted upon, regardless of whether it's something you feel is significant or not significant. This really highlights it. It's not really in the Haemato field. This shows you data from BRCA testing in the US. And basically, surgeons over there are actually acting upon, doing double mastectomies, taking over his house on the basis of BRCA VUS variants. Again, quite a worrying thing, and again, probably one of the reasons why we're actually going to go away from reporting variants of unknown significance. Again, depending on the surgeon how much work they do, patients who see more patients tend to do less of this, but in general, they all treat BRCA1 and 2 VUSs as pathogenic drivers, which again is worrying. So how can we address that? And probably the easiest way to address that is to actually look at data that we've all looked at. So this is the first round of the pilot myeloid gene panel that was sent out, and again, 27 labs did this. A variant in TEC2, a variant that we've seen before, and again, the 27 labs were split equally, really. 14 of these didn't report it, 8 reported it as VUS, and 5 reported this as pathogenic. The clues really are in the variant allele frequency. Again, what will put most people off is that it's actually reported in cosmic, i.e. it is being reported as being pathogenic. Also, it's been reported in DB SNP, as you can see here. That's the clue. It's been reported in DB SNP. So when you look at those, well, look at 3 of those 6 submissions, you see that the primary submission, when you actually delve deep into this, is actually a cell line. It's not actually a clinical sample, although it's been confirmed somatic, it hasn't. And these 2 subsequent reports refer to the first one. So there's a lot of that in Cosmic. So you put a single submission in, and everybody else refers to that. And that's how this has become quite a dirty database. You can see it's been reported many times in DB-SNP databases. Again, there was numerous other versions of this on the same send out. There are 2 TEC2 mutations there. Again, the first one reported pathogenic or VUS in 3 patients, and VUS or pathogenic for the second one in 13 patients. We see these very regularly. We probably see these 2, and they always occur together. We probably see them twice in every run we do. So they are normal SNPs. Obviously, they've both been reported in Cosmic, both confirmed somatic apparently. But again, these are normal SNPs. Again, in the same send out, there's also a very, very common well-characterized germline SNP in TP53. So the problem with all of this is that we're relying on a single suboptimal database for all our annotations. And again, I think that's probably what needs to be changed. So just 2 last slides, I'll only do 2. What are we doing now? Well, currently, and you can see this has got into UCAS in addition to scope, we are interested in looking at CLL and margin zone lymphoma, mainly because we've got a great interest in CLL. We've got Pete Hillman in the lab, we've got Roger Owen who's interested in Waldenstrom, so we've got a clinical interest, so that's why margin zone lymphoma is included. Ruth, I just noticed, has gone out, but she's also very much into Waldenstrom's macroglobulinemia and splenic margin zone lymphoma. So we've got a similar series. We'll run it on the same access array. This time we've got 33 genes. We've got 430 amplicons. The system works well. We've got TP53 there, which is a particularly large player because a large proportion of these requests are CLL patients about to start treatment. Have they got a TP53 mutation? Should we be treating them with BCL2 antagonists, etc.? So these are the questions we've been asked now. So we thought it was really pertinent that we ought to really get this within our scope. Again, the difference with TP53 compared to our myeloid panel is that we're looking at a larger proportion of the genes. So we're looking at exons 2 to 10 there. The long-term future, and this is my last slide, we're wanting to look at hybrid capture. We're wanting to actually do a large Haemonc panel, probably 300 genes. We've got proof of principle in this. We've worked with Philip Beer who was at the Sanger, he now works with us. He was the director of the 14MG. He had a very large panel, again, using SureSelect hybrid capture. The difference between that panel and what we're hoping to do in Leeds is we're actually not going to use Cavaris. Cavaris is a very expensive piece of machinery, very expensive to run on a routine basis because each sample is actually £5 for a little tube before you even start. We're going to use a fragmentation enzyme. This is a completely non-biased fragmentation enzyme. There are a couple on the market. Agilent make one, New England Biolabs makes one. This is a non-biased way of cleaving your DNA. Again, the other advantage is it also makes it applicable for paraffin-embedded material. Again, we'll do a conventional hybrid capture to try and cut down on the cost of this. We'll actually multiplex the sample and barcode them prior to the hybrid capture. So conventional hybrid capture, you capture individual patients. We aim to multiplex those patients, pre-barcode them, and capture maybe 8 patients at a time. So you are cutting down the cost, and the main cost for this type of technology is the hybrid capture and your baits. If you can make your cost divisible by 8, then it actually makes it now clinically, the utility more clinically usable. We're going to sequence it on an XSeq, and again, in collaboration with Glasgow University and Susie Cook, we're going to use their bespoke bioinformatics pipeline called Holmes. And I'll stop there, obviously I've gone over time. I'd just like to thank Catherine Cargo and Jan Taylor, who have been instrumental in getting this set up, Philip Beer for all his advice, Mike Short for being Mike, and Kathy really for being the head of the department. Susie Cook in Glasgow, we are doing continued collaboration with. Thank you very much.